Thank you very much, Dr. Matheson, and thank you for all you showing up. I can never imagine on the first day of the Chinese New Year calendar, I'm here rather than stay with my family. Happy New Year. So my talk is about hydrogen sulfide. Let me start this one with a short movie trailer in which I'm the consultant of this movie.
There is one thing that strikes fear in almost every man, woman, and child. While everyone does it, few will admit it.
Well, more bodily functions in the news this morning.
It wasn't me, that must have been them.
Yeah. Hey, Doc.
Mayor?
I totally just farted.
Some try to make it funny.
You know what I think about poetry?
I fart in your general direction.
Oh, hey, what the hell?
Some try to make it into art. But the truth is, it is something that most of us would rather not face.
Sorry, I farted.
Hello, my name is Albert Nuremberg, and I'm afraid of farting. But however, I have decided to face my fears right now, right here. Remember that hamburger you ate? It's still here.
Agricultural animals are farting us to oblivion. If we are to survive, we will have to face this head on.
What could be the biggest fart joke of all time? Our doom.
One man will go deep into the world of farts. Where do they come from? What are they doing?
Oh, damn!
This is the story that asks the question.
Who farted? The question is who hasn't? Cow, the most smelling element for the fart from a cow is methane. From a human being, it's H2S. About 2 or 3 months ago, when an article, and the feature article on the Global Mail in Canada, and talk about the story again about my team work. So I received an email from that one. It's from a person asking me, I have a problem, I cannot fart. That is after one car accident. I cannot fart. That huge, huge problem. And his belly kind of could be like this. He cannot eat, he cannot drink, he cannot sleep, he cannot even think. And then he asked me to see Dr. Wang, the fart doctor, can you help me to fart? Of course, I have nothing to do with it. So then I referred back to his family doctor. So this is a public perception of why hydrogen sulfide is related to our health. Many years ago, Scientific American asked me to write an article about hydrogen sulfide. I wrote this one. Toxic gas and lifesaver. When the article published, I remember vividly I had a banquet event in Ottawa. So I got an email. This email has come from a gentleman from Denver, Colorado. And the email started like, Dear Dr. Wang, with great respect, I read your article. And thank you, that article helped me to solve one mystery I haven't been troubled me for a long, long time. My wife, bracket, ex-wife, had a heart problem. In my household, the water supply comes from the well in my backyard. One day, my wife took the shower. The whole house is full of the smell of rotten eggs. When she finished the shower, she fell down, passed away. Even up to today, no doctor gave me the diagnosis and how my wife died. Your article told me that smell is coming from hydrogen sulfide, therefore, is hydrogen sulfide kill my wife. I want to get your legal advice to sue the company who drilled the well in my backyard. So I read that letter, I said, wow. I write back immediately, said, you have my full sympathy about what happened, but I really cannot help you about your legal advice. If you want my personal opinion, anything that smell though, that smell survived your wife for a long, long time, help your wife up to that day. Because the moment you still can smell that one, that is a totally safe range. Our nose is about 200 times more sensitive than the threshold level of hydrogen sulfide become toxic. So now you can see that's a public perception why hydrogen sulfide means nothing to our body except but toxic. So we started this study about to see how hydrogen sulfide really help our health. If you look at the literature, and up to this moment, I think this update, and yesterday I did while I was waiting for 4 hours in the Washington airport, and go to the Google Scholar. Currently, the top 5 mostly cited paper in hydrogen sulfide biology and medicine in the whole world, number 1, number 2, and number 4 is come from my lab. So, which is really showing the appreciation of what we have done to this field. Now, many people ask me how at the first place that moment, that inspiration, that spark triggered your interest to study hydrogen sulfide. So that is a reporter from Nature asking me the same question. And he interviewed me in 2009 and then published that 3-page of interview on the Nature. It started, let me just read your first paragraph. When Rui Wang saw the painted Easter egg his 10-year-old daughter had brought home from school in 1998, he was unaware of the stink they were about to create. The eggs were proudly displayed as objects of art in the family's glass cabinet. Then one day, says Wang, I came home, and the whole house was filled with a stinky smell. One of the eggs had fractured, and the distinctive whiff of hydrogen sulfide filled the air. At that time, my lab is focused on carbon monoxide, as well as nitric oxide. My believing is in our body, we should have many signaling molecules of gas, now we call it gasotransmitter, perform the similar function, but not only limited to the NO and the CO. So I thought there should be more of those gasotransmitters. That moment triggered the thinking that why not the hydrogen sulfide? The first thing we go back to the lab is to see whether our body produces hydrogen sulfide. People say, yeah, dumb. Everyone knows we produce hydrogen sulfide. But where? Is bacteria in our gut or actually in our cell? So to answer that question, this is our initial study. This is the paper I just mentioned, the number 1 and cited in the whole world in this field. We show at that time we cloned an enzyme called CSE in the cardiovascular system. The CSE uses the L-cysteine as a substrate or homocysteine to produce hydrogen sulfide, ammonium, and pyruvate. We demonstrate CSE enzyme is expressed in liver, mesenteric artery, tail artery, pulmonary artery, aorta, and also actually we detect the production of hydrogen sulfide in different tissues. So this is the first study. Almost 20 years later, we all know hydrogen sulfide in our body is produced by 3 key enzymes we call the CBS, CSE, and MST. They all started with either homocysteine or L-cysteine and produced this one as the main product rather than people said side product or waste product. When we discovered hydrogen sulfide actually is produced, now our question turned to why do we need it? Very often those molecules produced, they come and they go, doesn't play significant role. The first model we look at is vascular smooth muscle cells. So what we show here is this is a red carotid artery. And on this right side, we didn't do anything as a control. On the left side, the carotid artery, we use the balloon and to pass through to damage the endothelial cell layer to stimulate smooth muscle cell proliferation. So this is on the same rat. You can see after 1 week after that balloon injury, there's significant neointimal formation in this area and hyperplasia in this area. 4 weeks later, it becomes this big. If we give the animal injection of sodium hydrosulfide as a donor, of course, this is the pharmacological intervention. And 4 weeks later, you compare this animal, you can see, yeah, those neointimal and formation and hyperplasia of smooth muscle cell disappeared. This is the summary of those statistical analysis of this data. What this information tells us, hydrogen sulfide can stimulate, can inhibit vascular smooth muscle cell proliferation, because once you give it, you inhibit all these things happening. But the question is, this is not really a physiological thing, because we inject sodium hydrosulfide. How do you show this what happened in our body? Now you look at here, this is wild type mice. This is a genetic modified KO mice by knocking out that enzyme so that you eliminate H2S production from the blood vessel. Now here you can see, compare this area, and you can see this area is significantly enlarged. This is a summary which tells without endogenous H2S, smooth muscle cell proliferation is promoted because H2S play an inhibitory role. So that is a study we completed. And then we also isolated single smooth muscle cells from wild type and knockout mice without endogenous H2S. You can see this. In the absence of H2S, proliferation is promoted. Seems like a small study, getting interested. And then when we published that study as a second follow-up, and the Science News immediately put another news article said, wow, another major discovery. And Wang's team and other people showing hydrogen sulfide can really inhibit cell proliferation. That could give us a hint. What if inhibit cancer? What if inhibit other type of cells? So very optimistic that this provide a universal solution. Now we continue to say this may not be universal. Then we're looking at another cell type, which is endothelial cells. Now we actually demonstrate H2S in the endothelial cell is stimulate the proliferation, totally opposite to the vascular smooth muscle cell effect. Now here you can see this is the model. It's aorta, isolated aorta from a wild type mice and plated on the culture tissue. After a few days, there's a new endothelial cell formed tube generated. if we take the same tissue from KO mice without H2S, you can see that endothelial cell outgrowth inhibited. Even in the presence of VEGF stimulates endothelial proliferation and still shows significant difference in the KO mice. If we use another model, this one is a burning model. What we did is in the back of the mice, and we have a fire burning area. We generate this area. You can see this small area. And this in the white type of mice. On the day 15, you can see significantly recovered. But in the knockout mice, the recovery is inhibited. Now here's the summary. If this is white type, you can see recovery date very fast. But in the knockout mice, recovery date is delayed. Again, showing endogenous hydrogen sulfide actually inhibit, stimulate the proliferation. And without endogenous hydrogen sulfide, and the endothelial cells proliferation actually is increased. Oh, this labeling is opposite wrong. So now we show there's a two opposite effect of hydrogen sulfide. A single gas molecule can selectively modulate smooth muscle cell proliferation and endothelial cell and proliferation. So we thought this is a big deal. But then, again, there's another article in the Science on the May issue, again challenge the hydrogen sulfide story. They said with all those upcoming new discoveries, whether there's really a physiological meaning. Because physiology is the integrated science. It's not about isolated one protein, one cell, or three cells together. You have to show the function. The question is that what happened to the blood pressure, for example? Good question. So that's on the May issue. On the August issue of Science, we published the paper to tell the effect on the blood pressure. And these two mice, and tried to see their blood pressure level. The people said curiosity kills the cat. The mice are the same. But these two mice and become famous because one of them also go to the Nature and become their news article and picture there. So this is what we discovered. If we knock out CSE, that's KO mice, and the blood pressure will be gradually increased in an age-dependent fashion, and you can see that the age 12 weeks reached about 130 millimeter mercury. If we do the heterozygous knockout, which means knock out 50% of that enzyme, you can see the blood pressure increased by half, this wild type. 5 years work actually only generated this single figure and in the whole lab. That's kind of to my, I talked to my student, my fellow, the same things about if you think about your work, you have a quick turnaround, and usually yes. But if you want to really make a meaningful discovery, that's the lesson, 5 years, one figure. When that discovery published, and kind of interesting, public is very interesting to something directly linked to their health, especially this study linked to the fart. So instantly become international overnight news. More than 50 countries put that paper on discovery on their headline. In Canada, that become a headline story, the whole page of Global Mail, and talk about the discovery. And then there's some readers' comment coming in. And Global Mail and forward to me. The first one said, since the fart gas can deal with hypertension, my wife from now on will not complain about my presence. That gentleman has some issue in the kitchen. And then another one said, this must be a government conspiracy to cover up the stinky smell of Toronto. You see, where does that come from? Coincidentally, when that paper published, Global Mail put on the newspaper, the civic service people in Toronto have strike because the negotiation with the contract, salary increase. And that during the strike, Toronto, every week they pick up 2 luggage, Tuesday and Thursday morning. You need to put all your luggage kind of in the front, especially restaurant area. And you put it on the pedestrian pathway, those people come here to pick up. For a whole week, no one pick up the luggage. And the luggage piled up, the smell of the city is terrible. Then they say, so you just want to say the smell is good for our health. So that's kind of a story. How does hydrogen sulfide decrease blood pressure? Now, if you look at this one, it's Y-type endothelial cell staining of brownish staining or CSE protein. You can see the condensed in the endothelial layer, smooth muscle, and other areas you all have it. But endothelial layer is most significant. In our knockout mice, endothelial layer CSE is completely removed. What that happened? Make a long story short, we demonstrated hydrogen sulfide generated in the endothelial cells will function as to stimulate KTP channel in smooth muscle cells. When the KTP channel open and smooth muscle cell relax. Another function is for hydrogen sulfide would be it functions as EDHF, endothelium-derived hyperpolarizing factor, which has been a puzzle for 20 or 30 years. No people know what is the nature of EDHF. They all know EDRF, which is nitric oxide, endothelium-derived relaxing factor. So we demonstrate hydrogen sulfide actually is EDHF2 released from endothelial cell acting on the small to medium conductance KCA channel, and also the final result, the same relaxed vascular tissues. I don't want to show you the data, take my word for it, because this one probably composed of about 50 papers. So I don't want to show you one by one. Another story we talk about, if hydrogen sulfide is so important for the circulation, and then I want to give you the example of how it affects atherosclerosis. We feed these animals and with high fat diet and compare WT mice and KO mice to see their atherosclerosis development. This is from WT mice. This is their carotid artery. You can see smoothly linear things, there's no block. But if the same thing happened in the CSE knockout mice, and not only there's a lipid metabolism change, but they also, you can see, for example, in this area, there's a significant plaque formed. And that one, you can see once you have the blood flow through, definitely reduce the blood flow. Conclusion, hydrogen sulfide is so critical to prevent the development of atherosclerosis, especially in the early stage. The mechanism, of course, including many different things, including interfere with the lipid metabolism, and reduce the coagulation of platelet, inhibit smooth muscle cell proliferation, help endothelial cell to cover the surface to form a monolayer. All these things contribute to the protective effect of hydrogen sulfide on atherosclerosis. So I will quickly move this one, then right away go to another area, fundamental area, which is the mitochondria, rather than kind of you specific talk about cardiovascular system. Anyone can tell me what this photograph about? Mm-hmm. Say it again. Glowfly larva. Kind of you got it. I thought you will guess because I come from Canada. This could be the icicle in the winter time. Yes, this is the bacteria colony. This bacteria grow up in cave in Mexico, and actually this is a picture from National Geographic. In that cave, H2S concentration can kill the elephant. There's no life form should be there because it's so toxic. But these bacteria grow up there. They use those high concentration of hydrogen sulfide as their energy source. And there's also some beautiful and actually tasteful fish in that cave. The question is how the fish can survive, because all the water become hydrogen sulfide soup. But the fish beautifully there. Every summer, there's a tourists go there, and they want to see what happens. They wear the mask and have all those self-protection. And the ultimate goal is to taste that fish in that cave. Now, my question when I saw this one is, how does bacteria survive? Bacteria and us, we are all kind of fundamental life forms. Now let's look at the hydrogen sulfide metabolism in bacteria. And there's two types of bacteria. One is the bacteria use hydrogen sulfide. And another one is bacteria, they produce hydrogen sulfide. But both of them form a sulfur cycle, so the oxidizing and reducing and recycle to sulfur. And in the bacteria, their pathway for the transsulfuration is identical to human. But their pathway, we call how the hydrogen sulfide produced in our body, we call it the reverse transsulfuration pathway. The real transsulfuration pathway happened in the plant and in the bacteria. But the end result is still the same. They use the enzyme, give different name, but still CSE, they call it LDC. The end result is they produce hydrogen sulfide. Why they produce hydrogen sulfide? If you look at each bacteria, and they have on their membrane have lots of almost like our mitochondria, they have their reaction center. In this reaction center, what happened is the electron transport chain happened there. They use the H2S as the electron donor and coupled with hydrogen and gradient cross this membrane, ATP synthesis, and generate ATP, which is energy. And that happened in bacteria. They don't rely on oxygen, but they rely on hydrogen. If you have hydrogen sulfide, if you give the oxygen, you will kill this bacteria. Now the question is, what is the connection between bacteria and a human being? And Lynn Markley's theory, the endosymbiotic theory, is where the mitochondria come from. They come from bacteria. One eat another. That's why in the mitochondria you have a double layer, double layer. So this is essentially how the mitochondria formed. Now once you have kind of the mitochondria, you have those receptors, and probably you call it whatever, the iPhone or things. And then you have the transporter. Transporter give you all those nutrients, such as L-cysteine. You get into the mitochondria. Mitochondria will generate hydrogen sulfide and produce ATP. Then there will be beautiful story. If that's true, the question, why do we bother about the oxygen? You know, we have the heart attack. We have a stroke. All those issues because of oxygen. Without oxygen, ATP production will stop. Without ATP, your body is dead. It's all about energy ATP. Now, if we have this H2S-driven ATP production, why we even care about oxygen? Think about all the hospital emergency. You have a heart attack. The first thing is give you the oxygen. And this, if we can wake up this, I call it the ancient and the old mechanism for ATP production. If we can wake it up, at least we will save us a window time for emergency. Think about it. We just need to carry a bottle of hydrogen peroxide. And anywhere you go, if you have natural disaster, and you break your leg, you lose all the blood, you go to the war zone, and you don't have a blood transfusion coming in, give them hydrogen sulfide. Let them the ATP production continue. Save enough time, go to hospital, then you go back to the routine. That makes me excited. I said, wow, we should start this one. We should start this why we inherited the bacteria into our mitochondria, but we don't inherit that H2S-driven ATP production process. Now, you know in the mitochondria, there's a 13 gene, it's a mitochondrial gene. If you look at those 13 genes, they don't produce any enzyme which can produce H2S. But in the mitochondria, there's 100 proteins. Those proteins are coded in the nucleus, but then they transported into mitochondria to perform function. If there's any enzyme there produce H2S, of course, the first enzyme I'm looking is the CSE. Unfortunately, CSE is not expressed in the mitochondria. They expressed in the cytosol. So then finally, here is what we demonstrated. In the cytosol, CSE is located, but it have a pre-sequence can be recognized by the heat shock protein, which is the chaperone protein. When the calcium concentration or oxygen partial pressure decrease, TOM20 on the outer membrane of mitochondria will be assembled, form TOM complex. And the chaperone protein will go to fetch the CSE protein and bring it to the TOM20 complex. So that's the triggering factor and the chaperone and the moving from cytosol to the mitochondrial outer membrane. Because protein have a three-dimensional structure, the first thing need to be linearized, and with help of a TOM complex, get into the matrix. Now the enzyme come here to cut off a pre-sequence. Now you have a completely enzyme get moved into the mitochondria, and in the presence of HSP70, and it will take the three-dimensional form. Now it's become a functional CSE moved from cytosol to the mitochondrial matrix. Now the purpose, of course, is to move this enzyme from there to inside to produce ATP, or produce hydrogen sulfide. You may question why you bother H2S as a gas molecule. It should be perfused from, even produced in the cytosol. It should perfuse into the mitochondria if that's where its function would be. Theoretically, yes, it's membrane permeable, but we don't know how far it can go because there's a loss. Cytosol is not vacuum. It's a soup, all the protein soup there. Lots of things along the way will scavenge hydrogen sulfide. So hydrogen sulfide produced in the cytoplasm may not even be moved anywhere, will be just stay in the location. That's the first thing. Second thing is hydrogen sulfide production need a substrate. The substrate is L-cysteine. But then if you look at the L-cysteine concentration compared to cytosol and mitochondria, that's a major difference. In the cytosol, there's a cysteine concentration. It's about 1/3 of in the matrix. So cysteine cannot move across mitochondria membrane into mitochondria. So rather than that one, if you have an enzyme inside the mitochondria, you can fully use that 3 times higher concentration of L-cysteine to produce H2S right in the spot to produce ATP. We have demonstrated all those things experimentally, step by step. And then the final question, are you sure they really produce H2S, produce ATP? Now here is the experiment on the isolated mitochondria, and we measure the ATP content. Now this is normoxia, normal oxygen pressure. You can see when the oxygen partial pressure decrease, ATP production decrease. Once we produce hypoxia, which is 0 oxygen, now we start to give hydrogen sulfide. And you can see hydrogen sulfide at low concentration. And here without hydrogen sulfide, ATP level is here to 0. But then when you gradually increase H2S concentration, ATP production increase. Rather than conventionally people believe why H2S is toxic is because it inhibits ATP production, specifically interact with cytochrome C. That in the high concentration, that is in the presence of oxygen. Once you don't have oxygen, it produces ATP. People may also question that, compared to the oxygen, that cannot be even considered. Very small amount of ATP production. That small amount of ATP production saves your life. You don't need to kind of live in the rich oxygen area. You just normal oxygen. Here, you just need a small boost to save life. That mechanism is so critical. So that kind of back to many years ago when we final stage on we sent to the PNAS, and everyone happy, but the editor is not happy. I'll tell you the story. The editor sent back said, no, you use the mitochondria, isolated mitochondria to measure ATP, which is not physiological. And you already kill the cell. You take the mitochondria. How do you know that's the real in the real cell preparation still there? So I made the argument, a rebuttal. I said, the oxygen-dependent ATP production only happens in the mitochondria. No one can tell me outside the mitochondria where the ATP produced. If I have a mitochondria, I already demonstrated. The response is, doesn't matter you believe or I believe. If you don't have the whole cell data, I'm not going to accept. So you cannot argue with the editor. So we tried the whole cell again. We got the similar conclusion. So to summarize my presentation, and I hope by now you can be convinced hydrogen sulfide is so critical. It is produced in our body, I can almost say in every cell. Currently you can tell me that there's one type of cell, they don't produce H2S, I really want to see, tell me which cell. Almost every cell, I can only say almost. And it performs profound physiological function, affect almost every function of our body. The only thing is right now, we don't know. There are so many things we haven't even started, but everything you started, you see significant effect. And also that effect is not non-specific. It's very specific. As I told you, smooth muscle cell stimulate proliferation or inhibit proliferation. Endothelial cells promote proliferation. Even you think about the cancer cell, currently lots of studies in cancer, at a different type of cancer, hydrogen sulfide played a different role, either inhibit or promote. So it's very important, and in conclusion, hydrogen sulfide is a gasotransmitter, and we cannot live without it. With that, I will conclude my presentation here and acknowledge CRHR support, which is equivalent to the NIH here, NSERC equivalent to the National Research Science Funding NSF here, and the Hartz-Rue Foundation of Canada over the last many, many years. Without the support, we cannot do it. And also, I have to acknowledge the people in my team, and over the many years, going back, and there's about 122. I did the calculation, and over the last 20 to 25 years, and go through my lab. I cannot really mention. I tried to figure out to put up 122 trainees in my lab in one photo, and I failed, and therefore I decided not to single out any individual and the trainees. Thank you very much for your patience.
The mice where you knocked out CSE, is there any specific phenotype that you might have noticed? Are they just normal?
Hypertension.
Hypertension.
Hypertension is one, and also their body weight change. Currently, one study in my lab is doing it on obesity. We try to further to develop that one. The theory is hydrogen sulfide is a bad guy. It's a promote obesity. And because also in our team, we did before with my collaborator, Dr. Wu's team, and we show overproduction of hydrogen sulfide in the pancreas beta cell actually is the cause of diabetes. Genetically, if you overexpress CSE, you will produce more H2S. H2S kills the beta cell and also inhibit the insulin release. The similar story happened in the adipose tissue, which is fat. And so that could be also promote obesity. So I cannot really tell you the phenotype, but certainly we already know that body weight is a significant difference. And CSE knockout mice is lean and compared to the Y type.
But there isn't any mutations that have been associated with a particular disease in humans, that is.
There's no specific disease due to that one. But you think about Saul Snyder's lab in the Johns Hopkins, my long-term collaborator, and they published a paper in Nature 2 years ago to show the CSE knockout mice also developed that disease. What they call disease is neurodegenerative disease. No, it's not. No, also it's kind of a not common name, but they're also showing that one is due to the lack of H2S. It eventually develops that disease. But whether this CSE gene mutation is specific and kind of a one-to-one match to one disease, no. Could be facilitating factor. Yeah.
Can you reverse the phenotype if you give to the CSE knockout mice H2S?
Yes. Yeah. Yeah.
And the role of the sulfide in CNS is not known at the moment, or is that a completely different field?
So can you repeat again?
The role of H2S in the CNS.
Oh, it's no.
The circulation, right?
Yeah, because I'm not a neuroscientist, and certainly the early study in the Kimura's lab in Japan, they focus on the neuroscience. But in the brain, what happened is interesting. CSE is not a dominant enzyme, but CBS, another enzyme. Another enzyme in the brain, but to study the endogenous effect of H2S in the CNS system, in the neural system, it's not easy because the CBS knockout mice cannot survive for more than 4 weeks. While we are lucky when we have the CSE knockout mice, they can just live. But CBS, you cannot live. People try to generate heterozygous knockouts, but not pure. You can see there's something, but you cannot really demonstrate the rule. So that's kind of a problem.
So I was actually surprised about the phenotype of the CSE animal, because I thought that since you have 2 additional pathways to generate H2S, the MST and the CBS can compensate for the H2S deficiency on CSE animals, and then the phenotype might not show, right? So are these 3 pathways present all at the same time in your endothelial cells, or those are pathways, or those are mechanisms to generate H2S that they are different in different cell types?
There's a 2 part of a question you ask. One is whether the compensation by other enzyme can really kind of reduce the damage of CSE knockout. No, they cannot compensate. And in our CSE knockout mice, we measure the total H2S production significantly decreased, and very significant. In some organs, 0. See, for example, in the mostly in the kidney and in the heart, in the blood vessel, once you knock out, there's no H2S. The second question is interesting. You use the example of endothelial cell about these 3 enzymes. These 3 enzyme distribution is very cell type specific. Like CBS, in the endothelial cells, I still believe does not even exist. Last 2 or 3 years, there's a few papers come out to show CBS expression in endothelial cells. I read those papers, I'm not convinced. Although they publish, the evidence is not solid. Nowadays, as a student know, there's paper published, don't believe them. And you have to use your own eyes. But the question is MST. MST indeed expressed in the endothelial cells. But most of them are expressed in the mitochondria. So this MST is very unique in the mitochondria. But the role of MST, some people see it's very important, some people see less important. That's still wait to be seen. And even like if you go to the liver, in the liver, there's all 3 enzymes there, CBS, CSE, and things. But in many systems we tested, when we block all other enzymes, there's no difference. But the moment we block CSE, right away, the parameter changed. So it's still showing CSE is the dominant enzyme there.
So as a natural,
go ahead. No, we'll get to that.
Natural product chemist, we talk about garlic. That is sulfide compound. It has reputed health benefits. Do you link those to H2S production?
Yes. If you just take a clove of the garlic, and you just want to measure how much H2S is there, and you will see surprisingly nothing. There are no H2S. The trick is you have to add something. That is something getting into our body. The trick is the GSH. You have to produce the reducing condition. So if you want to measure how much H2S in the garlic, you have to add it artificially, add the GSH into your preparation. Then right away you can measure H2S. You can see the H2S production. So what that tells us, if you simply kind of smash the garlic and without intake, just garlic smell itself is not H2S. But when you eat the H2S garlic and the smell come out of your mouth, that is H2S. So now when people talk about a natural product, for example, using garlic or other things to provide H2S, it's not about the smell and the product itself. It's when the product gets into your body and generates smell.
So you presented in your in vitro endogenous experiments that H2S can inhibit vascular growth.
Vascular smooth muscle or endothelial cells? Which cell? It inhibits smooth muscle cell proliferation.
So how do you know if it is inhibiting the growth or promoting the apoptosis?
Complicated. We get kind of somehow not consistent result, depending on the tissue. When we use the human smooth muscle cell line, and we see mostly H2S inhibit, H2S affect apoptosis, but not about proliferation. But when we use the mouse smooth muscle cells, we see affect both processes. The reason, I don't know.
So is it known how the cells react to the exogenous hydrogen sulfide, especially the one in the human gut, there's like microbiome that producing the hydrogen sulfide, and how is there any like signaling between like how they signal the hydrogen sulfide produced outside the cell than the one that's inside?
Very hot topic. The interaction from microbe and the human body. Some of you or all of you follow the literature back to a year ago, and it became a hot topic in the science, in the medical science. It's by fit the obesity with the non-obesity people's bobo things, kind of your feet. And then what happens, they believe when you transport their content from non-obesity subject from their gut into obesity object, and then you will reduce the obesity. And the reason they said is moving the different colonies of bacteria. My interpretation is not moving the bacteria colony, it's moving hydrogen sulfide producing colony. You're introducing hydrogen sulfide producing colony, specific one colony from one group to another group. Therefore, you actually promote hydrogen sulfide. If that's true, I would say, for example, you move a group of bacteria which do not produce lots of hydrogen sulfide, and then move this one to the obesity objective, so that they will be, this colony will become dominant colony, will produce less hydrogen sulfide, then the obesity issue solved. That's back to my first hypothesis would be hydrogen sulfide actually promoting the obesity. So the interaction, it's actually the interaction between microbe and human and microbe and microbe. Because different bacteria colonies, they have a different interaction. Hydrogen sulfide could be like a chemical agent to determine when their interaction will, which direction should go.
Yeah, my question more onto like, is it known like how the cell responds if like the changing in the hydrogen sulfide that produced by, not by the cell, but by the microbe?
How the cell respond to the H2S generated by bacteria. And I really don't know how the cell can tell H2S come out from bacteria or come from themselves. H2S is H2S. Yeah.
Well, sorry, I have to ask this question. So how is that H2S promotes proliferation? How is that you think, I mean, proliferation is a very complicated signaling, a very complicated collection of pathways, and I understand the potassium pump and everything for your smooth muscle, but I'm not sure how to take it for cancer cells, for example.
Yeah, the same question is can also ask how hydrogen sulfide promotes the endothelial cell proliferation, right? So
okay, so indirectly.
Yeah. So this is all kind of, I can see there's a multiple target, molecule target of hydrogen sulfide, either in the cell cycle progressing protein or even in the nuclear kind of a coding protein. There's a multiple target. And I cannot really one thing for sure, it's not a single protein. It's not about hydrogen sulfide affect that one. That's where it happened about proliferation process. The research from both my lab and from many other labs, I think already identified multiple proteins to affect them. So which proteins dominate, which proteins not, I cannot tell, but it's just multiple protein affected.
Affected at what level? Stability level, transcriptional level, at what level?
If we talk about the individual protein, it can be any level. And also the literature says all different levels are affected. But we can see, for example, one thing, very important thing is at the post-translational level is the hydrogen sulfide-induced S-sulfidation. It's formed the persulfide with the sulfide group of targeted protein and then change the function. But that function is also happen, hydrogen sulfide affect even the coding process.
Is there any pump to pump out H2S from the cell?
No. There's also no pump to pump them in. There's a debate about gas channel. Some people said, let's use the analogy of a water channel. And it's occurring, all those things. They said probably occurrence should be the same channel to pump or to move the gas in and out. Very often they use the ammonium ion or ammonium gas. So far, I haven't found any solid evidence I'm convinced. I see the high top tier journal papers, they showing things, but they know not a single paper really shows the real is gas moved through those channels. They use different indirect parameters as to see this could be the case, but there's no solid evidence. And I can see the advantage, only advantage for the moving those channels is speed. If without speed, they will permit.
Well, the reason because I ask you is, as you say, if you have an attack or something, right, and someone gives you H2S, I mean, H2S has to get into the cell. And it won't be very easy for H2S to go through the plasma membrane unless it has some way of being internalized.
Extremely easy to go to blood-brain barrier. Extremely, yeah. And because it's a gas, it's easily penetrate the lipid bilayer. You think about the case they call the knockdown phenomenon, especially in the oil gas industry. In the oil gas, I come from Alberta, University of Alberta, where there's all the oil gas. And when you kind of take those natural gas contain high concentration of hydrogen sulfide. The moment hydrogen sulfide is released, you will be, before even you notice, you just sneak down, you're dead. And because the H2S immediately pass the blood-brain barrier to affect your respiratory center there, that interact with cytochrome C, stops the breathing. So it's very fast.
All right, for sake of time, we should wrap it up there and thank our speaker one more time.
Thank you.